Hi and Assalamu alaikum everyone. Welcome to my YouTube channel. So today's topic is really interesting as it's also related to my degree, that is bioinformatics. In this video we're going to discuss all about the pros and cons of using Linux or Windows as a bioinformatician, and which OS actually reigns supreme over the other. Of course, only we're going to discuss this only in terms of bioinformatics or their applications in terms of bioinformatics. Okay, so everything that I'm going to be discussing in this video will be based upon the knowledge that I have acquired over the last few years, or based on my own opinions with which you may agree or disagree. So without any further ado, let's get started. So, for most of you might not actually know what exactly is bioinformatics. So, before moving onwards to the main topic of the video, we're gonna see what it is. So, the easiest definition that I could actually come up with was that it is a field that uses computational methods in order to solve a biological problem. So, we're gonna be using all sorts of computational methods, like we're gonna use programming languages in order to develop some tools to help us in, like, if you want to predict the structure of the protein, we can use machine learning algorithms and stuff like that. We can develop tools that uses those ML algorithms and stuff. So it's like that. It is a field that actually combines biology, computer science, and also maths. The first reason that we are gonna see on why Linux is more preferred over Windows is that most bioinformatics tools are only available in like Linux. Most, but like not all bioinformatics software, such as softwares for structural annotation, variant calling, file genetic reconstruction, and some other softwares for drug designing, drug discovery, are all like written exclusively for Linux. Like, there are no such programs written for Windows or other OS. But there can be an argument that these softwares can be used on Windows. Alternative matters, like there is a online platform that is called as Galaxy, in which we can use bioinformatics tools such as FastQC, SAM tools, BCF tools, multi-QC, and stuff like that. These are all very integral tools in bioinformatics for like data processing, data manipulation, and stuff. But there are some like constraints, such as time. First of all, it's like when you run something on the Galaxy server, it takes like too much time because you are actually sharing it with many more people all around the globe. So it like take too much time. Last semester I did a project while using Galaxy, but it was such a mess. Something that would have been taken like uh, 30 minutes to max 1 hour on Linux actually took up 4 to 5 hours on Galaxy, or even more. So that was quite a problem. And then there is a problem of memory in like online platforms such as Galaxy. There you are only allotted a certain memory which you can use. And in bioinformatics, since you are like constantly playing with a huge amount of data, that limit is actually achieved quite early. And then you have like limited tool options. Even though there are so many options that we can pick in Galaxy, bioinformatics tools are generally so vast that Galaxy can't possibly integrate all of the tools. Basically, Galaxy is an online platform that integrates multiple and like various tools. There's like a vast amount of tools, but generally bioinformatics has way too many tools. So it is not possible for Galaxy to have all of them. But in Linux we can like easily use them. Another feature of Linux that is like way that makes things that makes actually life easier for bioinformaticians rather than using Windows is that you can easily create a biological analysis pipeline in Linux. So it's like a very good practice for bioinformaticians to actually automate their like genome assembly pipelines. It's like you can automate them from start to finish. If you do so, you would not have to like manually wait for one line to run and then manually start and then manually execute the next line. Since biological data is always very large, like more even like most of the time it's very large. So that would take like a whole lot of time, it would waste a lot of your time. But what you can do is that you can encapsulate all those processes in a bash script, and you can automate the biological pipelines via this way. Or there are many other tools available in Linux which you can use in order to do this, which are like not currently available in Windows. Okay, so another important reason is that Windows is like very slow at data processing. Many of you might want to disagree over here, but then again, if you guys have ever tried to open a FASTA file, it is like a special uh, file format for storing genomic sequences. If you guys have ever tried opening this file on your PC, on your Windows, via Notepad or any spreadsheet app, everything just crashes. It does not like work, it becomes very slow. It's like most of the time it does not crash, but it becomes so slow that you are like, oh god, where am I stuck? So that is because Notepad has a size limitation of 58 MB. So large biological files, these are not actually meant to be and cannot be opened directly with a text editor or spreadsheet app. Another reason is that if you're like opening it in a Notepad, you can't like peek at what's the header, what's the column. If we have the data in the column form, or if we have elements in tabular form, we can like peek in the data via Linux, that is via using the head or tail commands. These are like easily available in the command in the terminal of Linux. We do not need to do any special thing in order to get these. Another important use would be that in data wrangling we can like easily do that via Linux, and this cannot be like easily done via Windows. So that's a big difference. For example, we can use grep and redirect the output of any file to a different file. If we're like only interested in a certain part of that file, we can use grep. And if you want to like combine multiple FASTA files, so we can just use cat and then redirect that output to a new FASTA file. And these are like just normal commands that are already present in the terminal of the Linux, and it's like very very fast at processing all that stuff. Okay, so yet again, Linux has some more benefits over Windows. That is, it is free. You do not have to pay a single penny for getting any Linux distro. You can like get them for free. Linux is open source. Being open source means that you have all the code available. So what you can actually do is that this OS can be way more flexible for you if you're like a bioinformatician, or if you're more like in bioscience and stuff. There are already distros available that are mainly for the use of bioscientists, like biomed and stuff, and other distros like that. They already have some pre-installed tools, and they are like catered for bioscientists only, which means that and also if you want to like customize your own OS according to your needs, you can do that. Of course, since it's open source, you have all the code available. It is flexible. Another advantage is that it is like built-in programming languages. You do not need to install them, and even if you do, it's like just one command. You can install many more softwares and tools by using apt.yum, and that is like easier than installing softwares in Windows. So that's another benefit of it. So that was it for today's video. Hope that you guys understood why most bioinformaticians like prefer Linux over Windows. These reasons might not seem like way too big for some of you guys, but to us bioinformaticians, even the smallest of things count, like slow data processing speed and stuff like that, and like the availability of tools like grep, sed, awk in Linux. So these things help us in our work a lot, and make our work way more efficient. So that was it. If you guys have any recommendations, do share them in the comments below. So till the next video, Allah Hafiz.